Welcome to this explainer. Okay, today we are going on a seriously wild ride. We're going to travel all the way from the incredibly tiny microscopic processes of molecular biology, literally the fundamental building blocks of how life works, straight into the massive macroscopic world of high-tech automated engineering and robotics. We're diving into systems bioinformatics. We're going to see how biology acts just like a microscopic factory, and then look at how human engineers are actively reverse engineering that exact same factory. It is going to be fascinating.
Here is our quick roadmap for today. We're going to start off with the cellular computer, dive into the central dogma, and check out the immense power of proteins. Then we'll move over to the proteome, and we'll wrap the whole thing up by looking at how we're engineering the factory. Let's just jump right in.
Section 1, the cellular computer. We're talking about cells as literal information processing machines.
To really wrap our heads around life from an engineering perspective, you have to look at how living organisms act remarkably like tiny, super efficient computers. Think about it. While your laptop stores its source code in binary, just zeros and ones, biological cells store their code using 4 DNA bases, A, C, G, and T. And instead of organizing code into little digital files, a cell organizes its code into discrete segments we call genes. When your computer needs to speed up its processing, it copies its code into temporary RAM, right? Well, a cell does the exact same thing. It copies its DNA into temporary RNA. And ultimately, the software output of a computer is totally mirrored by the physical hardware output of the cell, which is the protein. And this beautifully twisted ladder is the actual physical hard drive for that genetic code, the DNA double helix. The rungs of this ladder are made up of nitrogen-containing bases, and they follow a really strict set of rules. Adenine, or A, absolutely always pairs with thymine, T, and cytosine, C, always pairs with guanine, G. This complementary pairing is brilliant because it's exactly what allows the cell to confidently back up and faithfully replicate its precious source code.
Moving right along to Section 2, the central dogma. This is where we get into the biological manufacturing process. So how does the cell actually boot up, read, and execute this source code? Biologists call this the central dogma, and it relies on a super elegant 3-part cycle. First off, the DNA code is transcribed into a very similar molecule called RNA. Next, that RNA gets translated into the complex language of proteins. And finally, those highly functional proteins are unleashed into the cell to do all the heavy lifting, including, by the way, replicating the DNA itself so the whole cycle can keep going for the next generation. Transcription is where the magic starts. Picture a giant molecular machine called RNA polymerase. It binds to a specific start address on the unwound DNA, which we call a promoter region. Now, it doesn't just blindly copy the whole genome. It's smart. It selectively targets exactly what the cell needs at that specific moment. Once it's locked onto that promoter, the RNA polymerase just zips along the template strand. It literally pulls in free-floating nucleotides from around it to build a brand new messenger RNA or mRNA strand. But there's a crucial difference here to keep in mind. In the language of RNA, thymine doesn't exist. Instead, the cell subs in uracil, or U, to pair up with adenine. So, the copying keeps going until the enzyme hits a biological stop sign, a terminator sequence. At that point, the newly formed mRNA just breaks off. This single, fragile strand is our temporary RAM. It safely carries the genetic instructions away from the highly protected DNA archive and heads straight for the cell's main manufacturing floor, the ribosome. Now, the translation step it's incredibly cool. The mRNA is fed through the ribosome and it's read in triplets. We call these triplets codons. So, for example, the start codon is virtually always AUG. And that specific sequence acts like a magnet, attracting a transfer RNA, or tRNA, that happens to have the perfectly matching anticodon UAC. Attached to the other end of that tRNA is a specific amino acid, in this case methionine. The ribosome basically ratchets down the mRNA 3 letters at a time, continuously matching up these tRNAs and linking their amino acids together to build a long polypeptide chain. Which brings us to Section 3. The power of proteins, the actual outputs of our cellular factory. Okay, get ready for a completely mind-blowing number.
1.27 times 10 to the 130th power. That is a number so unbelievably massive, it's basically impossible to wrap your head around. But this jaw-dropping statistic represents the staggering mathematical complexity and the potential variations of just a single, relatively small protein, once that amino acid chain folds up.
Let's actually break down the math behind that massive number. So there are 20 different naturally occurring amino acids. If you build a protein chain that is just 100 amino acids long, which honestly is quite short for a protein, the number of possible combinations is 20 to the 100th power. It creates an almost limitless design space. It literally allows nature to engineer a protein capable of performing virtually any biological function you could possibly imagine. Section 4, moving to the proteome. Let's talk about the challenge of functional complexity. As we pivot from the code to the physical output itself, we hit a massive wall of complexity. See, your genome is relatively static. The exact same DNA exists in your eye cell as in your liver cell. But the proteome is highly dynamic. It's the complete set of proteins being expressed by a given cell at a specific time, under very specific conditions.
And this poses a huge, huge question for biological engineers. Why on earth is mapping the proteome so much harder than mapping the genome? I mean, if we have the source code, shouldn't we easily know what the output's going to be? Well, it turns out just having the DNA blueprint isn't nearly enough to understand how a cell is actually functioning in real time.
The real answer lies in the dynamic modifications that happen after the code is read. Actually, scratch that. It's more like a single gene doesn't just equal one static protein. Alternative RNA splicing can stitch together different parts of the code to create multiple protein variants from the same blueprint. On top of that, you have post-translational modifications, which is just a fancy way of saying the cell adds phosphate, lipid, or sugar groups that radically alter how a protein works. Then you have protease cleavage, which literally cuts the protein structure, and dynamic non-covalent interactions, where proteins clump together to form massive supermachines. This is exactly why protein levels and forms change so drastically depending on the specific tissue type or even the environmental stress the cell is under.
Finally, Section 5, engineering the factory, how we are reverse engineering these cells using robotics and informatics.
To understand this mind-boggling level of complexity, traditional test tube biology just doesn't cut it anymore. We absolutely have to move to the macro world of modern systems bioinformatics. The very first step is capturing the genes we want to study. But cloning genes from eukaryotic organisms, like humans, is immensely difficult. Our genomes are vast. The human genome has around 22,000 genes, and even a tiny microscopic worm like C. elegans has 19,000. Plus, our genes are broken up by intervening sequences called introns. That means we can't just copy the DNA directly. We actually have to extract the mature mRNA from the cell and reverse engineer it backwards into complementary DNA, or cDNA, just to get an uninterrupted, usable sequence. To handle this at the scale we need, engineers have developed the high-throughput cloning pipeline. First up, they select target genes by mining massive computational databases, like RefSeq. Second, they amplify those exact sequences using PCR, polymerase chain reaction. Third, they capture those genes inside a circular piece of DNA called the plasmid vector, which basically acts as a biological delivery vehicle. And finally, they rigorously sequence and verify the clone to make absolutely sure no errors sneaked in during the copying process. Because the scale of this pipeline requires thousands and thousands of reactions, humans are literally taken out of the pipetting loop altogether. We use liquid-handling robots with specialized addressable tips to dispense reagents into 96-well microtiter plates. This automation is a game changer. It completely eliminates human error, allowing scientists to run thousands of cloning reactions simultaneously without breaking a sweat.
Of course, robots are really only as good as their programming. Behind all this hardware is a massive database brain known as a Laboratory Information Management System, or LIMS. Systems like FlexGene track the specific barcodes of every single 96-well culture block and every 48-sector agar dish in the lab. They track the lineage, the physical location, and the biological validation of tens of thousands of clones in real time. This combination of heavy-duty robotics and deep informatics has completely, fundamentally revolutionized how we study biology today.
But you might be wondering, why does all this matter to you and me? Why are we building these massive robotic factories in the first place? Well, it's because the vast majority of successful therapeutic drugs are directed against proteins, rather than against genes or transcripts. By mapping this super dynamic proteome and producing these biological machines in the lab at scale, we actually gain the ability to design life-saving drugs that precisely target and interact with the faulty mechanics of a specific disease. It's absolutely vital work. We have seen how cells process information just like computers, and how we're now using high-tech robotics to reverse engineer those very cellular factories. It leaves us with an incredibly exciting frontier to ponder. As our macro labs get more and more advanced and our understanding of this microscopic source code becomes even clearer, what entirely new biological software will humanity write next? Thank you so much for joining me on this explainer today. I really hope you leave feeling inspired by this incredible intersection of biology and engineering.